Hello everyone, I am Dr. Anil Kumar Kalvala, currently working as Research Associate under the supervision of Professor Mandip Sachdeva, FAMU. Today I am presenting my work entitled with Transcriptomic and Proteomic Analysis revealed cannabidiol and tetrahydrocannabivarin signatures to overcome DOX resistance in MDA-MB 231 xenografts in athymic nude mice. Here we have hypothesized that CBD and THCV, minor phytocannabinoids, can overcome the doxorubicin resistance in MDA-MB 231 triple negative breast cancers. Herein we aim to study the potential molecular targets of CBD and THCV in attenuating tumors by RNA sequencing and proteomic analysis. Briefly, we injected CBD 10 mg per kg and THCV 15 mg per kg IP to DOX resistant 231 tumor bearing mice, and at the end of the study, the tumor tissues were isolated and conducted RNA sequencing, proteomics, immunoblotting, and RT-PCR studies. More importantly, this was the first study to investigate tetrahydrocannabivarin's anti-cancer potentials against resistant cancers, as well as the first study to conduct a systematic analysis, identification, and validation of potential targets using RNA sequence and proteomic data of these two drugs' anti-cancer potentials against DOX-resistant triple-negative breast cancers. Followed, we measured the IC50 and combination index of DOX, CBD, and THCV in 231 resistant cancer cells. These are the IC50 values in 2D and 3D spirit cultures of DOX, CBD, and THCV respectively. We also calculated a combination index in which CBD or THCV were found to be synergistic against cancer cells when combined with DOX. The 231 DOX resistant xenograft model was developed by injecting 2.5 million 231 DOX resistant cells in a 1:1 ratio suspended in matrigel into the right flank of athymic female nude mice. When the tumor volume reached 1500 mm3, the animals were treated for 2 weeks with DOX, CBD, THCV, and DOX in combination with CBD and THCV. After 14th day post-treatment, DOX plus CBD at P less than 0.01 and DOX plus THCV at P less than 0.05 combination treatment group substantially reduced the tumor volume when compared to control. Additionally, gene expression studies were performed in MDA-MB 231 DOX resistant tumors following CBD and THCV treatment. When compared to normal control tissue, RNA sequencing suggested differential regulation of numerous genes after various treatments. The determination of differentially expressed genes between normal control and treated tissue was done using a heat map which demonstrated that 1050 genes were upregulated and 1280 genes were downregulated after treatment with CBD, and 980 genes were upregulated and 10,010 genes were downregulated after treatment with THCV. The linkage of biological pathways was determined using the cake pathway analysis demonstrating differentially elevated genes. According to cake pathway analysis, differentially expressed genes after therapy were found to be engaged in many pathways including metabolic, immunological, inflammation, mitochondrial function, apoptosis, and RNA transport. Among these, AMPK pathway, PDL1 pathway, NF-kappa B pathway, NLRP3, STAT3, TGF-beta, and apoptotic pathways draw our attention because of their significant role in chemotherapy resistance against breast cancers. Following proteomics analysis is performed in the same tumor tissue homogenates, the expression of proteins matching the RNA sequence data was discovered. In this study, proteins such as protein XRP2, transcriptional enhancer factors, and AMPK were upregulated, while hemoxygenase 1, SOD, BCL-XL, P38 MAP kinase, PDL1, CD133, NF-kappa B, catalase, tenascin, SP1, and NLRP3 inflammasome were downregulated in CBD or THCV treated with DOX. We also validated the RNA sequencing and proteomics data in tumor tissues using Western blotting and RT-PCR. All of the proteins that were upregulated and downregulated in RNA sequencing and proteomics were reflected in our Western blots and RT-PCR. The interesting thing here is that when CBD or THCV was combined with DOX treatment, H3K4 methylation and H2BK5 acetylation were significantly reduced. The majority of these proteins were linked to histone acetylation and methylation. This data suggests that these numerous proteins were involved in doxorubicin resistance, and CBD and THCV have overcome this resistance by regulating these resistant markers at the gene and protein levels. The overall conclusion of this study was that CBD and THCV could overcome doxorubicin resistance in MDA-MB 231 xenografts in athymic nude mice. H3K4 methylation and H2BK5 acetylation were identified as potential resistant markers against doxorubicin in MDA-MB 231 tumors, as well as the molecular targets of CBD and THCV, which is quite interesting and was reported for the first time in this study. Furthermore, literature supported that all upregulated and downregulated proteins were linked with these histone modifications, which collectively have a tight control over resistant tumor progression and development. However, more molecular and knockdown studies are needed to better understand the role of these molecular targets in the resistance of MDA-MB 231 breast cancer cell lines to doxorubicin. We would like to thank RCMI, the U54 program, and the Center for Health Disparities for funding this research, as well as our postdoctoral and doctoral students in the lab for their hard work in making this happen. I would like to express my gratitude to FAMU and RCMI for providing this wonderful opportunity. Thank you to everyone who is watching this video.